a membership where you pay like $6,000. Me and my mom and dad are all.
All right. So the first part of cell respiration is glycolysis. What is glyco? Sugar. And what is lysis?
Breakdown.
So that's what glycolysis is, splitting of sugar. Alright, so what happens in glycolysis is we start, like I said yesterday, we start with glucose as our model because it starts at the very beginning. And so we're just starting with glucose here. A lot of other things can be used in the entirety of cell respiration. We can start other places. I haven't talked about it yet. So again, these pictures are in your book, and I really like it. And hopefully y'all have learned by now to study, like really using the figures and diagrams to help you study, have you? Because they help. The one in your textbook, I guess, looks a little different.
Yeah, so it looks good. There's like a second square.
Because there's 4 stages.
Oh, no, why only 3 here?
Because this is the old textbook. I've always talked about 4. I just noticed the new textbook has the other box. Anyways, okay, so we're talking about now, stop. You cannot talk and listen to me at the same time. That's rude. Thank you. So again, the point of all of this is to make some ATP. So some of the stuff I put in here is not in your textbook, like this, so you should write it down or listen. So how does glucose get into the cell? Using glucose is a polar molecule, it's a sugar, so it can't pass through the membrane. Generally, it's going to go down its gradient into the cell. It goes through a protein, a membrane protein. It's actually called GLUT1. Anyways, that picture kind of shows how it happens. So once glucose is in the cell, it might enter glycolysis, or it could be used to do other things for the cell. Starting point for building other organic compounds, whatever. But let's say that it goes into glycolysis.
Is it facilitated diffusion where it uses the phosphate?
That's active transport. Yeah. Facilitated diffusion is facilitator is someone who helps. Diffusion is down a gradient. Did you just hear me?
So this protein doesn't require anything to move it.
A facilitator is someone who helps things, like a protein. And diffusion is going down at something's gradient. So facilitated diffusion is going down a gradient using a protein facilitator. Does that make sense?
So the facilitator.
You do have a test on that tomorrow. Yes, hopefully you remember that. All right. So glycolysis, breaking down or splitting sugar. And actually what happens in glycolysis is split into 2 pieces. It's important to count the carbons. When we go through this whole process, it's important to count the carbons. So notice glucose is a 6-carbon compound. Therefore, it's a 6C. At the end of glycolysis, you have 2 3-carbon compounds. Have you lost any carbons?
No.
Not at all. They're also there. They're just put into 2 different molecules. Glycolysis is an evolutionarily the oldest way that organisms can make ATP. Let me go back up to my coffee. Like I said before, all organisms do that. Prokaryotes, they evolved first. Yeah. No, I actually have 2 copies. It's got white on it. What do you do?
The other one right there.
Okay. The problem is you only make 2 ATP molecules for every glucose. That's not very much. The entirety of aerobic cell respiration can make how many?
36 to
36-ish to 38 maybe. That's a lot. So there's only 2 ATPs. That's not a whole lot. The thing is, all life, like the first cells were prokaryotic cells, and they had no membrane-bound organelles. They were unicellular. They didn't need a lot of ATP. That was enough to sustain them for a long period of time. So, why does it make sense that glycolysis takes place in the cytosol? Prokaryotes didn't have organelles. So it makes sense that all they had was the cytosol, so it takes place there. Good. So also, it also makes evolutionary sense that glycolysis evolved first because early Earth had no atmosphere, had no oxygen, okay? Aerobic respiration doesn't make sense that evolved early on because there was no oxygen at all. So it makes sense that glycolysis is actually anaerobic. You don't need oxygen for it to happen. So the cells had to, you know, now when I say the words had to, they didn't have to do anything. It's what natural selection sort of over time, they were selected for, and therefore they survived and had more offspring. It's whenever glycolysis evolved, it allowed what we call an evolutionarily selective advantage. All of a sudden, these cells that had the ability to extract and make this ATP molecule from a glucose, well, they were more able to survive. They therefore reproduce and pass on their genes to their offspring. Does that make sense? So not to say things like they had to do something, well, they didn't have to do anything. Just what natural selection did over time. Anyway. But it makes sense that it evolved without the use of oxygen because there was no oxygen. There we go. It's important to know that all organisms on the planet use glycolysis. Everything. All right, so there are a bunch of steps to glycolysis, 10 to be exact. And you do kind of have to know what's going on in them, but you don't have to memorize all of them. In your book on page 168 to 169, it goes through all 10 steps. The diagram I have on the board is in here, but at the bottom, like whoa. Okay, you see all of them? When you do your talk drawing, you're going to have to do all this. You're going to have to talk to me about all this. Okay, but you don't have to memorize all this per se. What you have to memorize is kind of what's on the board, and generally kind of what's going on. You don't have to memorize every single enzyme. There's only one enzyme in all of aerobic or cell respiration theory you have to memorize. It's called phosphofructokinase. It's in here. It's only because it's a major one that's regulated to help coordinate everything. But we'll talk about that at the end of the chapter. Otherwise, you don't have to remember anything in terms of the enzymes right now for this class. Take biochem and other stuff in college. All right. So there are 10 steps, and we separate into phase 1 and phase 2. The first phase is what we call the energy investment phase. What that means is that ATP is actually used. 2 ATP molecules are used during these first 5 steps. Now, why do we talk about ATP is actually used? Well, how does ATP actually work in the cell? Make things phosphorylated, and when things are phosphorylated, they're usually relatively unstable. It's like it's holding a hot potato. You want to let it go. So the reason why ATP is used in these first 5 steps is to make the glucose molecule unstable and more reactive and more likely to split. So by the end of the 5th step here, the glucose molecule is actually split into 2. We call B3P molecules. All right, so we'll go over those in a little detail in a minute. The second set of 5 steps is the energy payoff phase. And notice over here, we actually make 4 ATPs. But what's the net total ATP production? 2. So -2 plus 4 is a +2. Yes. We also make some NADH molecules. NADH, remember, is the little NAD plus is a little bus that carries those little high energy electron tumblers in hydrogen, right? So NADHs are produced, and if there's oxygen, and if you're in a eukaryote, it'll move into the mitochondria, drop those electrons off of the ECC, where they can be used to help generate more ATP. If you are a bacterial cell, that NADH is not used for that. It has to be converted back to NAD through the process of fermentation. We'll talk about that at the end of chapter 2.
So it's like glucose gives you energy, and like steady eating gives you like a good source of energy. But like, why is that? Like if you drink like Red Bull or soda, you get like lots of energy and like
Okay, so that's caffeine.
Or like is that? Oh, so it's not like.
It's not this.
Oh, okay, never mind.
Yeah, you have sugar.
Like sugar?
It affects your nervous system. It's a stimulant.
I love sugar. Like coffee.
Like a honey candy.
You can get a sugar high, and that's not this. You don't use, all that sugar you eat in M&Ms is not immediately being used to make ATPs. A lot of that is being used and converted to glycogen or fat, to be honest. You don't need, having all that physiologically is complicated, but it makes you feel like euphoric, but then you crash really fast. So, anyway, that's a little different. That's not this at all. Yeah. We'll do the nervous system later. Okay, so I just talked about this. Now, y'all need to pay attention. This is the first 5 steps, just a different diagram than what's in your book, and I'm going to talk about them. But again, you need to know the overall goal of the first 5 steps. It's to make the glucose unstable and to split it into 2 pieces. So I'm going to kind of go over these, and you can watch. Some people, some of you, I've taught a lot of y'all in the past, and some people like me, I like to see what's going on in structure that helps me understand why this is happening. And so that's why I go over this, so you can actually see. And this is all in your book, too. But so we start with a glucose molecule. Here it is. Now, again, they're not showing the hydrogens and the hydroxyl groups over here. They're there. They're just not showing. So in the very first step, ATP is used. It phosphorylates this guy. See that? Okay, we're on our way to becoming unstable. The enzyme that does it is called hexokinase. I'm going to talk right now, but a kinase enzyme, have I talked about a kinase enzyme? You need to know. Any kinase enzyme phosphorylates things. A kinase enzyme phosphorylates things. You need to know it eventually. We have a whole chapter 11. That'll be completely and utterly brand new to all of you. If you can know something already before we start, it might help a little bit. But kinase enzyme phosphorylate thing. Hexa means what? You might know 6. The 6 carbon sugar, hexokinase phosphorylates the 6 carbon sugar. A lot of times it's easy to memorize this stuff. You actually understand where the word comes from. Anyways, okay. So we phosphorylate it, make it a little bit unstable. We use our first ATP. The second one here, this enzyme is called an isomerase enzyme. And basically what it does, it breaks this thing down and puts it back together, making an isomer. Anyways, you don't mess that. The third step, we phosphorylate it again. So notice this is between the third and the fourth product here. Notice this thing looks super symmetrical, doesn't it? Yeah, okay. It's really unstable. It's got 2 phosphate groups, and it's ready to be split down the middle. So this last step here, we split this guy into 2 pieces, and you make 2 isomers of each other. You actually make something called dihydroxyacetone phosphate and something called glyceraldehyde 3-phosphate, G3P. This dihydroxyacetone phosphate is converted to G3P, and the only thing that moves on to the next 5 steps is to G3P. So this one has to be converted. But anyways, by the time you're finished with the first 5 steps, you have 2 G3P molecules and you use 2 ATPs. That's what you need to know. Yes?
What did you say was converted into G3P?
This little guy, this thing is split into 2 pieces. One piece is called G3P, the other one is called dihydroxyacetone phosphate. The isomers of each other, G3P will move on. This dihydroxyacetone phosphate has to be converted to G3P, so it can move on. Okay. Yeah. Yeah. You okay?
Yeah, okay.
I'm going to mention right now, have you heard of G3P before? Did you know that the direct product, what is the product that put us in the system?
Carbon dioxide.
The direct product of what comes out of the Calvin cycle. You may know. It's not a glucose molecule. You're welcome. It's a G3P molecule. The Calvin cycle doesn't actually make glucose. The Calvin cycle makes G3Ps, and then 2 of them put together will make a glucose. So, anyway, we'll see G3P again. It's important to also notice the crosstalk between the stuff. So G3P is actually the direct product of the Calvin cycle. Anyway, yeah.
Like, how many molecules are made? Like, is there ever a where they're not?
Yes. Actually, the G3Ps don't have, in photosynthesis, the G3Ps don't have to come to make a glucose. The G3Ps might be used to make something else, like it's through carbon sugar. So, yeah. Questions on the first 5 steps of glycolysis? Yeah.
So the 5th step, aldolase splits glucose into 2 isomers,
Yes.
and then the dihydroxyacetone and G3P.
Yes. No, you don't have to memorize all that stuff. Take it back. The only things you need to know, like I said, you start with the glucose. 2 ATPs are used to make it really unstable. It splits into 2 3-carbon sugars. Both of them end up becoming G3P, which moves on to the next 5 steps.
So what's the step below that? NADH?
Yeah, that's step 6, and we're going to get there over here. So hold on. This is the energy investment phase. Okay, notice we use energy, yeah? We didn't really get anything in return yet at all. Okay. So the next 5 steps, there's a lot of energy payoff, a lot. Okay, so we're going to talk about it. So like I said, the G3Ps, and notice over here this little arrow goes like, it's because this isomer is converted to G3. Anyway, so they're showing 2 of these going down. In your textbook, this diagram, let's see, yeah. Does anybody have your book?
Yeah.
So on page 169, I want to point something out, because when you're actually looking at it, you get confused. You see that number 2 right there? Yeah, I just want to point it out. It's like page 169, 168, 169. So if you look at those 10 steps, look at the boxes on 168. Notice they're just little boxes. But notice as you move from 168 over to 169, you see there's a 2 now by the boxes. Because there's actually 2 of these things going through. But your book doesn't show 2 sets of them, it only shows 1 set, but they use that little superscript of a 2 to indicate there's 2 of them. But sometimes you will miss that, and you're like, I don't understand. Okay, so just notice that in your book. Again, you're gonna have to draw all this stuff and do it all, so you'll be referring to this a lot. Y'all need to bring your textbooks to class on Monday and Tuesday and Wednesday, so basically every day next week. Okay.
Can we introduce y'all on this?
Yeah, you can. Yeah. I think it's easier to use a textbook, but it's fine. That's me. You can. I don't care.
Not easy on the back, though. It's very good idea.
All right. Hey, y'all. Tomorrow's the last day to donate to babies that can have tetanus. Have any chain will pass this around. I emptied our box yesterday. She gave us a new one. She's like, people are actually donating. She was like really happy. So let's show her how happy we are. I'll put money in there too. Okay. So G3P, what happens first is that we extract, or this thing becomes what? So we're taking electrons off of here and hydrogen, so the G3P becomes oxidized or reduced.
What?
G3P becomes oxidized. Okay? So notice an enzyme goes right up through phosphate dehydrogenase, you don't need to know that, but an enzyme comes in and facilitates the oxidation of G3P to produce this thing, 1,3-bisphosphoglycerate. You don't need to know that. But notice, we made an NADH molecule. We actually made 2 of them, 1 for each G3P.
Wait, something has to do with it.
At the end of third.
Do we have lunch after this?
You go to after fifth. Have fun.
What? We have a
Or actually, no, you don't have a break. You don't have a break. Sit down. They're leaving.
Oh, where is that?
They're going to that thing.
I'm gonna go ask Ray.
No, I
didn't know we were doing.
Oh, yeah. They're doing that question and answer thing. Have fun.
Hey, ask Greg how in the world you have to realize how.
No, I don't know.
Yeah, exactly, and ask how ready you feel to be a role model.
How do you feel about feeling, I would have being a celebrity with like getting hours and I'm like.
Should have worn your prom shirt that you gave us last year. You're like, Mr. Smooth, like, this is like.
Yeah, like be like. It was literally my 5 minutes. I think I'm sure. I'm sitting there.
Okay.
Come listen, hi.
I should ask about, so what Wait, was it this way?
Alright. I'll keep talking. You know, I'm gonna finish psycholysis and then the rest of the time I'll just let y'all be. I'll just stop.
I love it.
But I need to finish this. Okay. So we made some NADH. Now if we are bacterial cells, then that NADH really isn't used for anything. Okay? But if you're a eukaryote and if there's oxygen, the NADH will move into the mitochondria and help make some more ATP. But it just, at this point, we don't have to worry about it. So the next step, notice we take off. Now remember, there's 2 phosphate groups on these little guys. So one of them will come off and make ATPs. So we would make our first 2 ATPs. And then, what? Did you ask something? I wanted to point out something, actually, while or whatever. We use 2 ATPs up here, yeah? So notice there's 2 phosphate groups. I want y'all to look at something. Because when y'all do your chalk drawing, you're going to get like really confused. I'm going to point it out now though. Notice in this step, so when you split this guy, how many phosphates should be on each one? One. But notice how many are on this? 2. What happened? Notice in this little step, a phosphate came on. You see that? All right, so this is what we call an inorganic phosphate, that little PI. The enzyme that takes the electrons off at the same time it pops the phosphate on. I'm just telling you what happens, okay? So that's how each one of them has 2. All right, anyway, so in this step, one of these phosphates is taken off. It's this one here. So we make our first 2 ATPs. And then we're being isomers, making isomers. We lose the water here. Okay, and then the very last step here between what we call phospholynyl pyruvate, PEP. We're going to come back to this later too. Something called pyruvate kinase comes in, takes that last phosphate group off, makes an ATP, and we're left with what's called a pyruvate molecule. We have 2 of them. The carbons are all still there, so there's 6 carbons still. Thank you. Have we made NADHs, we made some ATP, thank you. And that's about all. Oh. Questions? I'm not finished. There's a whole PowerPoint.
There's a whole different.
No. I mean, this PowerPoint goes on for a little bit. So, don't worry about this. Now, I need to talk about this substrate level phosphorylation. I'm actually going to rewind for a second. Go way back here. All right. So, ATP, notice at the bottom is made in glycolysis. Some ATP is made in the Krebs cycle, and a lot of ATP is made in what we call ETC, oxidative phosphorylation. All right. So, the way ATP made over here is actually called oxidative phosphorylation. But the method in which ATP is produced in the Krebs cycle in glycolysis is very different. Okay? It's called substrate level phosphorylation. So I'm going to explain it now. A substrate is what an enzyme acts on, yes? Okay. So what happens, let's focus on this very last step of glycolysis. Here's our enzyme pyruvate kinase. This is a little purple thing. Okay, it takes in PEP, which is this guy with this phosphate group, and it takes in an ADP. You see that? The enzyme grabs onto the 2, changes shape, breaks off this phosphate off of here, puts it on the ADP. ATP is produced and released, and pyruvate is released. Substrate level phosphorylation. All it means is the enzyme directly transfers a phosphate from one substrate to the other. That's all it means. The way the ATP is made in the ETC, and it's through an enzyme called ATP synthase. Remember that from Bio 1?
Yeah.
Very different. We'll come back to it later. A lot of times, all of this won't actually make sense until I finish talking about it all. And then you're like, oh, because we'll continue to make connections. Yeah.
You said the enzyme directly transfers
A phosphate off of one substrate going directly onto ATP.
From phosphoenol.
Well, in this particular case, but it happens, remember, here too.
Okay.
Substrate level phosphorylation also happens in the Krebs cycle, so it doesn't have to be happen. Okay. And these enzymes that do this phosphate transfer are kinase enzymes, like I said before. Okay. So again, we do all this accounting as we go through all the stages of respiration. But we used 2 ATPs, we made 4, so our net gain is 2. We also made a little bit of NADH, we made 2 of them. And we started with 1 6-carbon sugar, we ended with 2 3-carbon sugars. that's the kind of detail you need to know for this. It's not terrible, it's really, compared to all those 10 steps. Okay. Yeah. That's just a picture again. This.
So those are
The pictures?
enzymes, is that like?
These are what all the enzymes look like.
Oh.
Yeah.
Yeah.
Can you see, look at the coils, those are the alpha helices?
Yeah.
And there's beta pleated sheets in there, which are these things. These are computer generated images of what the enzymes look like.
So like, if there's billions of cells within our body, how many enzymes are there?
Oh man. I don't even know a lot. A lot. Okay. So that's not all. So for a very, very, very long time, 1 billion years or more, this is how life on Earth survived, but it's okay because they were just bacterial cells and it doesn't matter. They didn't need a lot of ATP. So if there's oxygen present, then there's a lot more to be done. The last thing I want to talk about, I'll mention here is, let's imagine you're a prokaryote. You're a bacterial cell and you don't have mitochondria. Well, then what do? The only way you can make ATP is just to keep doing glycolysis. That's the only way you can. But in order to keep doing glycolysis, you have to have more glucose. You have to have ADP. You have to have some of these inorganic phosphates, and you have to have an NAD+ molecule, right? Glucose should be pretty readily available as long as the cell's not starving. It wouldn't be hard to make the ADPs because the cell's going to use the ATPs pretty rapidly and convert them right back. Okay, inorganic phosphates should be pretty readily available as well. The problem is just NAD+, because you make an NADH. Where is it going to drop its electrons off? It's got to get rid of the electrons and the hydrogen. Okay, if there's no oxygen, where does it do it? Well, it's through a process called fermentation. Okay, there's 2 ways: ethanol fermentation and lactic acid fermentation. There's actually more ways we'll talk about. But I think I'll just stop here because I think I made a whole PowerPoint on sterile fermentation. I'll just wait. You can use the rest of the talk studying. Your louder
My phone was stopped working. I can't share it until we start the comment out.
Yeah, no.
Yeah.
And also, I don't know how to do, like, there's only code that's now on the USC application says you have your recommenders use the comment out.
Whenever sending out 44, I need to do that. Yeah, I don't like the coalition thing, to be honest.
I don't know. It's just issue.
On the coalition,
Yeah.
I don't know anything about that.
I mean, you don't even mention it, the idea of it was just to observe the holiday, so you want to do it a lot.
Hold on.